Good afternoon. It's wonderful to see so many of you and so many young people. It's wonderful being back in a university. I spend a lot of my time talking to politicians and public policy people, so it's great to talk to young people again. My talk today is about genetically modified crops, and my plea really is, let's give these crops a try. They are no means a silver bullet. There are other problems in feeding Africans, but let's not prevent Africans from access to this technology. You may or may not know how important agriculture is in Africa, but these numbers aren't that important, but the importance is that the amount of grain we have to import. So we actually need to do better. A lot of it is lost due to post-harvest, and many countries depend on agriculture for foreign currency. Now, I always start with this slide because I'm in awe of plant breeders. I had a meeting with some of graduate students just now and told them how important plant breeders are in this whole thing. Can you imagine farmers looking at something like that and getting to that in 9,000 years? It took a while, but... and then that, and this. So, I mean, plant breeders are amazing. But sometimes we forget that nature doesn't give us everything we need. So we sometimes have to tickle what nature gives us by improving it, if you like, or changing it through mutagenesis. So we have to use radiation or chemical treatment. And there are many crops that have been developed in this, but nobody will know if they have harmful byeffects because none of them is tested. It's only genetically modified crops that have been tested for food safety, and they are tested as toxic, as if they were toxic. So they are subjected to toxicology testing, and that's pretty different from anything else. Now, I don't have to tell you this because I'm often not speaking to breeders, but people misunderstand the role of hybrid crops. So, they often say, oh, the poor farmers, they've got to buy their seeds. Well, if you plant hybrids, you've got to buy your seeds anyway. And a lot of people don't realize what goes into developing the lines, the inbred lines that are used to cross together to get a hybrid. And of course, if you plant your own seeds, of course you can plant your own seeds, but you're going to lose the traits. So you're going to lose whatever hybrid vigor it was. So that's a very important slide for my non-scientific audiences. But do we remember what happened when hybrids were introduced in the 1930s? There was huge resistance, similar to what is now against GM crops. They wanted to plant their own seeds. It was only drought in those years that showed that these hybrids could help them. Now most commercial farmers plant hybrids and buy seeds every year. And even in Africa, smallholder farmers, which we have the choice, they can either buy open pollinated varieties, or they can save their seed, or they can use hybrids. And they have that option. And many of them go for the hybrids. So it's not as if by saying GM crops are a good idea that you are closing the door on these other options. These other options remain. So, when they say, oh, the multinationals will take over in Africa, why? Farmers are not stupid. Farmers choose what's best for them. There are two main ways. I'm sure you all know this, so I don't have to tell you this. Two main ways for transferring, either using agrobacterium or microprojectile bombardment. And the agrobacterium, I'm sure you all know about it. And the particle gun uses... one of the earliest, I think I actually saw at Cornell, one of the earliest varieties of that actually looked like a gun. And they used gunpowder to shoot DNA on inert gold particles, and it lands up integrated into the nucleus or into a chromosome by recombination systems that happen. Now, of course, we don't rely on that to such an extent because the agrobacterium system is much more specific. But there were, in the early days when I started working in maize transformation, agrobacterium didn't work. So we did rely on the gun. But now we can rely on both if we need to. But we can certainly use, that's what it looks like now, far cry from a gun. Now, what I tell people who don't understand these things is that the source material for this, for the gun or for agrobacterium, is callus tissue. So that's undifferentiated plant tissue. Now, a bit like you get on the heel of your foot. But if you take a cell from the heel of your foot, you're not going to culture it into a human being. And animals don't do it as well. Plants are totipotent, as you all know. So plants have the genetic capacity to regenerate from a single cell, and then every cell will have that same DNA in it. So if you have the callus, you give them the right hormones, they'll produce fruits and roots, and you can get transgenic plants. And this was the first transgenic plant we produced in our lab, which was a maize plant that was resistant to a herbicide. And as I will show you, I didn't think that herbicide was important in Africa. How wrong I was. You learn on the job in this field. So some years ago, we had a meeting talking to African agricultural experts, saying, what are the crops, what are the improvements in the crops you would like that could help us through genetic engineering? And I'm going to come back to this, but in fact, I'll come back to it. So one of them, Africa is home to viruses that you've never even dreamt of in other countries. So I'll talk about that. We have weeds you've never even dreamt of. Obviously, we have the same insect resistance, but then we also have African varieties and cowpeas and other kinds of crops that we are indigenous in Africa that we want to. I'm going to tell you a very interesting story how links fungal toxins with insect resistance. This nearly fell off the bottom of the list because drought tolerant was seen to be too difficult and too multigenetic. And I'll show you that perhaps that isn't the case. And then there's a colleague of mine working on bananas, bacterial wilt. Then there's also vitamin-enriched crops. Why do I say not golden maize? You know about golden rice? Because in Africa, on the whole, and certainly in South Africa, if you are a black person, you will not eat yellow maize. Yellow maize is for fodder. White maize is for animal food. So if you want to produce beta-carotene in maize and make it yellow, you're on a hiding to nowhere. There's nitrogen use efficiency, which I might have talk about, and there's stacking. Listen to this. Please read it. You only use the GM approach if no other, cheaper, or any other solution is available, believe me. Because, as you will see, this has been a long process for me and my colleagues. And if there were a cheaper, quicker way, I would use it. I wouldn't go the GM route, largely because of the regulatory issues that are involved and the implacable opposition by a lot of anti-GMO groups. So let's look at them in case. Now, this is my research. So I work on maize streak virus. For obvious reasons, it's called maize streak virus. I do, as you will see, a lot of my work in Kenya, and this is a shamba, which is the Swahili for a small farm. And this poor farmer is not going to have much of a yield, both due to the virus and due to drought. So that's why we work on both. Look what it looks like. I wouldn't be saying it if it hadn't worked. So this is some of our plants which are non-GM, looking pretty miserable. And there's the result. Pretty good. There's a non-GM, there's a GM. Now, this is important. We were very thrilled when we published this paper, the title of which says Maize Streak Virus Resistant Transgenic Maize, a First for Africa. It was a first for Africa. It was a first transgenic that would be done entirely by Africans for a disease that's only found in Africa. But then, we made the headlines front page, which is always nice. This came out in Science. And we were thrilled about this because it says the first genetically modified crop developed entirely in Africa is gearing up for field trials. Its success will be a milestone. The date was 2007. We still have not done field trials. Why? Because it's simply too expensive. We're in partnership with a small seed company. A small seed company based in South Africa, which does export to other African countries, but they don't have this kind of money. It costs too much, largely because, remember, this is novel. We'd have to do all the rat feeding trials to make sure that this protein isn't going to kill anybody or harm anybody. So we have to do that plus all the field trials. Panar, our seed company partner, said, sorry, it's too expensive. And so that's why I say you need a good idea plus perseverance. Fortunately, I have plenty of perseverance. But so Panar was bought out a couple of years ago by Pioneer. And the agreement is that Panar can still market in Africa under Panar's name. So you'll still see in the farmer's field Panar, but they have access to the germplasm of Pioneer. So they have increased ability in their breeding programs. But, and this is where we got very excited, they have money for research. So we thought, wow, now they have money for field trials. Do you know what they said to us? This problem is only an African problem. African farmers haven't got enough money for us to recoup the expense it's going to cost. So sorry, it's going to stay in the lab. It's going to stay in the glass house. So maize streak virus resistance and linked ones that we've developed are staying in the glass house for the foreseeable future. However, my perseverance has paid off because we've got another project on drought tolerance, but I'll tell you about that a little later. So, herbicide resistance. Remember I said I didn't think weeds were a problem in Africa. Look at that. That is beautiful. But try and be a farmer and deal with that. That is a parasitic weed called witchweed or striga. When the maize germinates, it sends out chemical signals to say to the witchweed seeds, which are sitting there for millennia just about, until a maize germinates. There's your food. 40% of your food comes from us. Grow. Intertwine your roots with ours and we'll feed you. Talk to farmers. They say, if there's witchweed in the field, forget about maize. We can't grow maize. So it's a problem. So my organization, the African Agricultural Technology Foundation, that was in the introduction, please remember it. I'll be coming back to it. It's a terribly important organization for Africa. It was founded to transfer intellectual property, whether it's seeds, whether it's DNA, whether it's mechanization, whether it's know-how in seed companies, royalty-free, from multinationals to African farmers and seed companies to develop any technology that AATF thinks the farmers need and the farmers tell us. I was the first chair of the board and I'm still very involved with it. So what we did was we got an organization called BASF and they had developed a Maize-Pure, that's a herbicide, a Maize-Pure tolerant maize. This is not a GM product, and it's important for you to understand that. How they bred it is a sort of a slight mystery, but at any rate, here they are. So if you coat the seeds, you don't have to spray. If you coat the seeds in the herbicide and you plant them, then they are resistant. If they're not, the witchweed will grow. And this is a major success. So, but look at the result. Two things happened as a result of this. We first discovered that farmers said, gimme, 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 obviously. And in Africa, we do not have a big network of seed companies. So one of the things that the ATF and AGRA and other organizations have been involved in is helping seed companies develop so that they can do that. The other thing was, and this was certainly in the early days, we gave farmers bags of maize coated with the Maize-Pure. So the farmers would plant it. Now, many farmers in Africa intercrop with beans or legumes of some sort. So if they plant them and their hands are covered in the Maize-Pure, it kills the beans. Headlines in the Nairobi Nation, biotech maize kills beans. Simple problem, simple solution, wash your hands. But we had to get the message out. So what the ATF did, it got together with NGOs and put out little brochures and said, go into the fields and explain to the farmers they have to wash their hands. Simple, but very effective. And so we've been able to, because it's a non-GMO variety, we've been able to spread this and get our NGO network working. Insect resistance, you all know about this. I'm not even going to bother about it. You get that. The solution is Bacillus thuringiensis. You all know about that. Yes, yes, yes. Yes. Yes, right. We started it in South Africa some years ago, and this was one of the slides taken of, this is the non-BT growing madly trying to produce bolls, and this is the BT, and etc. But here's the difference in Africa. You see, a farmer who has to spray with insecticides will often mix his insecticide in a bottle like that. And you say to him, what are you going to do when you've finished? No, we'll just throw it on the ground, and then kids can pick it up and put water in it, and insecticide poisoning. You need water. In Africa, water doesn't always come out of taps. You have to collect it. And who collects it? The women and the girls. One of the other things that I'm involved in, I chair the Organization of Women in Science in the Developing World, and I'm giving a talk tomorrow about that. So if anybody's interested in Hooray for Women in Science, come to my talk tomorrow. And what happens if the wind blows into your face? Then you can have insecticide spray poisoning. Okay, but what I wanted to show you in Africa is the link between insect resistance and fungal resistance. If you're a farmer, and most of the farmers, many of the farmers are women, and you store your maize, you harvest your maize crop, you store it in cribs like this, and then the rain comes down and the sun comes down. If there are holes due to insect nibbling, this is what it looks like. And that can go things like liver cancer. That's an extreme case. But again, if you're a woman, you look at that maize and you say, I'm not going to feed that to my family, but I'm not going to throw it away. So what do I do as an African farmer? I brew it into beer. So the fungus grows and grows, and the mycotoxins. So in parts of South Africa, esophageal cancer is endemic. So that's just a link. And then in Africa, we've also got cowpeas. So my organization, the AATF, has got the BT gene, and we have transformed it with colleagues elsewhere into cowpea. And we are doing field trials in Nigeria and Ghana and a few other places. But of course, it's very dangerous, so it has to be fenced. So these field trials are all fenced. And of course, if you're a small little boy or a little girl in Africa and you see a fence, what do? You climb over it. So these fences have to be policed. So hence the expense, and so it grows. Drought tolerance, yes. Oh, I don't know if this has worked. No, it's just that was supposed to show the spread of drought in Africa, but it hasn't quite worked. Anyway, we all know that there is drought in many parts of the world, and with climate change, we are going to get worse. This year, our maize crop was down by 1/3. And I can't see it getting much better. But what we decided when we got involved in this project, I must tell you how I got involved in this project, because my work has always been funded by non-companies. So I don't have, except for PANAR, which helped us mainly in kind, certainly no multinationals have been involved in my funding. So it was a trust that funded the virus work. And when it got to the stage where PANAR said, look, it's going to stick in the glass house, they said, well, that's the end of our funding, quite rightly. And so then they said to me, and what are you going to do now? You can't just stop. I said, no, I'm going on to drought tolerance. And one of them, who might not have been the smartest in the pack, said, but what if there isn't a drought? And for the first time in my life, they said they'd seen me at a loss for words. But anyway, so what we decided to do is, fortunately, I was telling the plant breeders how important they are. The other group that is terribly important, but it's not sexy, are plant physiologists. Because without plant physiologists, we don't know what these genes are going to do to the whole metabolism of the plant. So they're terribly important. I'm very, very lucky because I share an office with a plant physiologist. So I said to her, Jill, where should we go to source our genes for drought tolerance? And she says, well, there's this interesting plant called a resurrection plant. And when it's hydrated, it looks like that. When it's dehydrated, it's dead, to all intents and purposes, except it's not. It's lost all its chlorophyll. It looks dead. But if you add water within 72 hours, it resurrects to that. So it's a bit of a miracle plant. So we decided to work on this, pretty stupid because, nobody knew anything about its genetics. So we decided, but first of all, we had to find the plants. And so one of my former postdocs left science to run an indigenous plant nursery. And so I said to her, Rachel, do you know this plant? It's called Xerophyta viscosa. Oh, yes, she says, it's in our catalogue, but nobody's ever bought it because it only flowers for 2 days a year. And who's interested in that if you're a gardener? She says it grows in cracks and rocks in the mountain, often close to water. But you can imagine how dehydrated that can get. So off we set. She said, I think they're in the Drakensberg. So off we set, and we climbed the Drakensberg mountain, and we found them on a rocky outcrop like that. And I was telling the students just before that it was kind of a rite of passage for every postdoc or graduate student in my lab to climb the mountain, dig up a plant with permission of the maize board, of the plants board, because this is an indigenous plant and you can't just mess around with indigenous plants. So you had to have permission, and they brought they're big plants, they bring them down in their backpack and they put their name on it in the glass house, and that's their work. Unless, unlike one of mine, he decided to see how it's responded to cold stress, and he put it in the deep freeze and went away. And the next morning his plant was dead. But he was a good student, so I let him off. But one of the times when we did this, we actually, I had a very good plant tissue culture person, and after about 3 years she learned how to germinate these plants, so now we don't have to do this. But 1 year we went up, and there'd been a heat wave. So you don't work in centigrade and I don't work in Fahrenheit, so it's 42 degrees centigrade, so most of you know what 40, yeah, it's hot. And then the night before we climbed up, it rained. And it was cold and misty when we set out. And when we got to the sort of stretch of rock that you have to cross with looking steep down on either side, it was below freezing. So it gone from 42 to below. And when we saw them, they were resurrecting. You could see these plants coming up. It was fabulous. But on the way down, I said to one of my graduate students, I said, "You pleased you came?" So she said, "Yes, but I'll never come again." So it was quite tough, but now we don't have to, which is a bit of a pity. But anyway, so I wouldn't be telling you if it didn't work. So this was one of the first genes. Oh, by the way, I was funded now by the Rockefeller Foundation. I was very lucky because I landed up at Davos at the World Economic Forum on a panel with Gordon Conway, who was the president of Rockefeller. And he was asked, this was 2002, this is, I'm really very old. And this was 2002. And he was asked, what is Rockefeller's next project? And it was after golden rice, and he thought, well, golden rice is finished now, it's all ready to go. We'll do something new. And he said, drought tolerant crops for Africa. So I patted him on the shoulder and I said, "Hello, Gordon, my name's Jennifer. Now, can you please fund me?" So he did. And so I was fortunate in that. But one of the people I reported to was very keen that we try for genes that were very novel. So one of the genes that, and this is the one that I'm showing you here, a single gene, is a membrane protein that has 7 transmembrane linkages and has no known homology to anything in the database, and has signal domains on the surface, so it transmits. But what we've done now is we've got 3 different genes. By the way, I have PhD students whose whole thesis says this gene does not confer drought tolerance on maize or on a transgenic. So, we went through a number, and not all of them worked. But we now have 3, and we use the promoter from this particular gene because that promoter is a stress-inducible promoter, so that it is only switched on. And interestingly enough, because of this plant living where it does, it responds to heat and cold, a little bit to salinity, and certainly to dehydration. So that's, this is some other plants showing the transgenics, the T3 generation. In Cape Town, you cannot grow maize to tasseling stage unless you do it in a conviron. And so it's very expensive. So when I worked with the Rockefeller Foundation, they were funding a lab in Nairobi And so a lot of our work is of the glasshouse work and the field trial work. Well, sorry, we haven't done field trial. Glasshouse work is done here in Nairobi at Kenyatta University. So that's been a very, very happy relationship with them. But it's a bit of a commute from Cape Town. I did it for about 6 months, 2 weeks Cape Town, 2 weeks Nairobi. I wouldn't recommend it, but it certainly got the work on. That's what I talk about, perseverance. But we also needed funding from the Rockefeller, so that was useful too. Okay, so this is the opposition. It's not opposition, as I used to say to my students, but this is the WEMA project, Water Efficient Maize for Africa. Here's the African Agricultural Technology Foundation, which is the broker. Monsanto gave us the gene. They were very happy to give us the gene. It's a bacillus cold-sensitive protein driven by a constitutive promoter. And so they gave it to us. CIMMYT, which is the International Maize and Wheat Breeding, has a home in Nairobi. They did the breeding into local varieties. And then we use the National Agricultural Research Services in the countries to test them. And so I used to go around talking internationally as chair of the board of AATF about WEMA, and I'd come back to the lab and my students would say to me, hey, but you're talking about the opposition. And I say, actually in science there is no opposition, because in the field we don't know what's going to work. So everything is open to until we test it. And that's why I'll say, let us test it. Don't say you can't test it because it might be dangerous. Anyway, I must tell you about field trials too. When we were starting to do the WEMA field trials in South Africa, they called me I was being chair of the board of AATF to go up to this little town. And the little town is called Lutzville. It's a tiny little town up the west coast of South Africa where it never rains during the maize season. And that's why they don't grow maize there. So it's a good place to do a field trial, because there's no competing maize that you could cross-pollinate or anything. So it was marvelous. So we went up there, and obviously it was wonderful. And I came back, and about 2 weeks later, a friend called me and said, have you seen the front page of the Cape Times today? Floods in Lutzville. So you see, field trials you have to do in multi-locations. And then there's the bacterial resistance. This is Xanthomonas wilt, which is a problem. How long have I got? Because I'm just rambling sort of on. I guess you have 15 minutes. 15 minutes, okay. Now I want discussion, so I'll try and finish in 5. Okay, so this is another one, and this is a colleague of mine, and she's gotten it to work. And what I always say, and I'm really a bit disappointed today, because her name is Lena Tripathi. She works for the IITA in you know her, in Uganda and Kenya now, and I nominated her for that L'Oreal UNESCO prize that I won. And I thought she'd be a shoo-in, and I heard today she hasn't gotten it. And I'm going to look very closely at who wins, because when I won, I happened to know somebody on the board, and in a drunken moment she told me that they nearly didn't give it to me because I was working on GM crops. So I'm going to look very closely to see who wins from Africa. They better be a bloody good scientist to outcross the... Anyway, so this, but you see the beginning there, Lena Tripathi, is I learned maize transformation from the experts. She developed it, or her group developed it. And she's gone into field trials. I mean, it's mind-blowing. Anyway, okay, now I have to end a bit of the controversy, because that's what's inhibiting things in Africa. You know, I used to be told that we have enough, the world produces enough food. Yeah, but the problem is poor physical infrastructure, etc. So we need in Africa any technology. And what I say to the Europeans, and you're not Europeans, but I mean, I say this to the Europeans, you've got double standards. Since mad cow disease, you can't feed animals with offal from the abattoir. So you have to feed them with soybean or maize. And because it's ineffective to grow them if they're not GM, you import them. But you don't allow your own farmers to grow it. You import them happily. In the early days of GM, they used to say to the farmers in Namibia who export cattle, we aren't going to import your cattle or your food or your meat if you feed them with GM maize from South Africa. But now they suddenly have to, so they've changed their tune. And I really, really have trouble understanding the logic. And it's not as if this technology hasn't taken off in the developing world. You know, it's now, this is my organization, ISA. And so this is the developing world, and that's the developed world. Let me tell you a little bit of South Africa. We've had commercial planting since 2000. 81% of white is GM, stacked with BT and herbicide tolerance. 93% of yellow maize. We don't segregate GM and non-GM maize in South Africa, and it hasn't affected our exports one iota. But this is work done by a friend of mine at the University of Pretoria. You see, South Africa is quite an interesting case study for smallholder farmers, because we are the only country where smallholder farmers have been growing for 8 seasons, 9 seasons more, GM maize that they eat. You know, it's not cotton, it's not soybeans that goes to animals, it's maize that they eat, as well as feeding to their animals if it's yellow, but the white they eat. So looking at the farmers, you know, they are taking it out. One of our biggest problems was getting the companies to sell it in small enough packages, because these are smallholder farmers. But, you know, one of the things he did is he looked at how popular herbicide tolerance was. Because I thought it's cutting down on weeding, and it's mainly women who do the weeding. And he asked them, if you had the choice and you only could choose weed resistance or insect resistance, which would you choose? I thought it was a no-brainer. Look at the insect damage. Weeds are always there. Insects aren't always. If we had to choose, we'd rather have herbicide resistance tolerance, but can we have both? So most of them are stacked. Kenya, I want to just talk about Kenya for a minute. Their imports were stopped. They were importing and doing field trials in Kenya. They weren't growing them themselves yet, but they stopped in 2013 because of the study that was done by a group under Seralini in France that purported to show that he said that the 90-day toxicological rat trials at high doses, which was standard to treat these compounds or foods as if they were toxins, that isn't long enough. You must actually do it for 2 years. Stupid old farmers, stupid old companies, or stupid old scientists who've been doing it for 90 days. It should be 2 years. This variety of rats has been bred so that it'll get tumors at the end of 2 years. It's supremely sensitive. That's why these studies are stopped at 90 days, so that anything that is vaguely abnormal will indicate something seriously wrong. And no GM product that's ever been looked at has shown anything problematic. And so, you know, the strains get 40% of the treatments had no controls. The use of accepted protocols, flawed statistics. Eventually, it was withdrawn. But although it had been headlines in our newspapers, the fact it was withdrawn was not headlines. So the Minister of Health in Kenya was a cancer survivor. And she went to the president and she said, we don't want anything more to do with GM crops, no more imports, no more growing, no more anything. And so the president stopped. He didn't even ask his biosafety committee, which he has. So that's been, now what's happened now, that the minister, the vice president is a man by the name of Ruto, and he is an agriculturalist, and he has said this is crazy. We're going to allow imports, and we're going to allow GM trials, and the first thing is going to be BT maize. We're going to allow its release in a few months, and then a few months ago he said we'll allow it in October. Watch this space. It's very difficult for a politician to say, oops, I didn't mean October, I meant January next year. So it'll be very interesting to see what happens there. Oh, and this is some of the farmers who came. This was actually published in the Nairobi Daily Nation in May this year. As Kenyans stall over biotech farmers, South Africans have happily adopted it. And these are just some quotes from farmers saying how much they've... Now, this is serious. This is Greenpeace, eh? If you go into Africa and say, if you eat this, you will become fertile. And that's what's happened. I spoke at the United Nations in 2003 or something. And Kofi Annan had invited us to speak, a couple of us to speak on GM crops in an open session. And they said openly, you know, Europeans, particularly the Scandinavian countries, have been telling African politicians this. So, you know, that's very difficult to come to gainsay. Sorry, that's food safety issues. Okay, I'm not going to go into that. I do believe in environmental testing, because what happens in South Africa might not happen in Zimbabwe and might not happen in Namibia. So you've got to do field testing. That I totally agree with. The food safety ones, we can. I totally agree with allergenicity. Do the 90-day test, but it's terribly expensive. So in 2011, GM crops have had fewer adverse effects on the environment than non-GM crops, because I'm just going into one of them, conservation tillage. I have a very good friend in South Africa who's a commercial farmer, moved to GM, and he says we go into conservation tillage, because before conservation tillage, before GMAs, you had to get rid of the weeds before you planted. So you tilled, and you allowed the weeds to grow up. Then you sprayed to kill them, but then you have to get rid of the spray. So you just wait until that spray dissipates, and then you lose your topsoil to the wind and the rain. Now you plant your maize, you let them grow up, you let the weeds grow up, and then you spray, and then the weeds can actually help as mulch. So conservation tillage, etc., have been pretty useful. So, ladies and gentlemen, in conclusion, they are not a magic bullet, but let us test them, let us try them. What we need is improving infrastructure. You know, when I first went to Nairobi, the potholes are so enormous. I was telling them just now that, you know, camels could drown in the potholes in the rainy season. You know, there are no roads and rails systems for transporting them. But most important, if you want to feed children, educate their mothers. That is the single most. They'll have fewer children, they'll have knowledge about nutrition, and that's the way to do it. So and wouldn't that be nice? So my take is that they can help to feed hungry people if farmers are allowed to test them. And I say, and this particularly for my audiences in Europe, shameful if well-fed Westerners using issues including trade to prevent this technology from helping to feed the poor. Thank you very much.